My name is Anna Capria. I work on the BV-BRC project as an outreach coordinator and bioinformatic analyst. I work at the J. Craig Venter Institute, which is one of 4 sites that are part of the BV-BRC. We'll talk a little bit more about that as we go on, but this is a 5-part series where we're just trying to reintroduce you to some tools and services within the BV-BRC. This really will serve as an introduction to the resource and give you some good background. The goal for our outline today, tell you what is the BV-BRC, tell you a bit about the tools we offer, tell you about how to make a workspace, how to organize that, and how to search for data within our resource. So I want you to leave this session feeling comfortable with what the BV-BRC is in general, and get you comfortable loading, storing, and retrieving information within the BV-BRC. So our mission is to provide a one-stop shop for data about human pathogenic and related bacteria and viruses, integrated into a scalable resource for comparative bioinformatics, large-scale integrative data mining, multi-scale systems biology exploration, and machine learning that will support the evolving needs of our infectious disease research community and serve as a paradigm for research data resources. And how we kind of embody that goal is when we start thinking about our key features. So we support a diverse bacterial and viral community. We have many different pathogen genomes and tools to analyze that data, which I'll tell you a little bit more on the next page. But by having this unified data model where we're adding metadata and other value to the data, then that allows you to integrate with our tools or other tools that you're using to do your complex analysis. Our efficient data management supports exponential growth of pathogen data. As I was putting together these slides, I had to update numbers from just a few months ago, and we had millions more genomes added just in a couple of months. So our database is growing every day. We do data polls nightly, and so if you check the numbers, they might be different the next day. And the data is pulled in with accurate annotations and try to make those consistent across different inputs. Like, for example, we have a project working on host name annotation, which will make it easier to search by host type because researchers across the world will put in different things, meaning the same host. So that's one of the ways that we try to add value to the data. We have automated and manual curation of metadata, which is sort of what I was just touching on, and want to make modular, interoperable, and compute-intensive, high-throughput data analysis services, and those we'll spend a fair amount of time talking about today, but really trying to reduce the computational burden on your side and allow you to run those high compute-heavy analysis on our tools and servers, which is all secure and able to be used by anyone for free. Integrative analysis of multi-omic systems biology data with visual analytic tools, phylogenetic and epidemiological analysis for outbreak response, some explainable AI and machine learning-based tools in the future, private user workspaces, which we'll talk a lot about today, programmatic and batch access with APIs in the command line interface. So if you're doing large data analysis, please let us know and we can point you in the direction of those tools. That'll be really useful if you're a little bit more programmatic in your practices. And then we do outreach and education like this webinar. So our user community is robust. We have over 44,000 users with 25,000 unique visitors per month, over 20,000 citations, and that they range from basic researchers all the way up to bioinformaticians and computational biologists and all of those things in between. So you can see our users growing monthly and citations. So I touched on our data, and we have over 11.8 million microbial and viral genomes. Most of those are viral. 8.4 million of those are SARS-CoV-2 genomes, and over 1 million flu segments, 32,000 bacteriophages, 10 eukaryotic hosts, And we have these uniform annotations across genomes using Figure-4 for viruses, which allows for us to annotate genes, proteins, and mature peptides, and other genomic features to make that data more valuable. And then the curative metadata, which we've touched on, and epitope, protein structure, omic data, microbial and host pathogen data, surveillance, serological records, and all of this data is aggregated from public archives like GenBank, UniProt, PDB, and from programs that we participate in from NIAID, like the SEERS and SysBio functional and structural genomic programs. We derive data from internal pipelines and do some data integration, and then you can also bring as much or as little private data into your workspace. So if you're running sequencing projects and you have a bunch of genomic data that you'd like to analyze within the BV-BRC, you're more than welcome to download that into your private workspace, and that stays private to you. We never see that. It's just sitting in your folder like a Google Drive folder if you're familiar. But we'll go into that a bit more. We've talked a lot about the data now. Now I want to just touch on our services at a high level. So we have analysis tools like assembly and annotation through BLAST and phylogenetic analysis, primer design, genome analysis. We'll look at all of these on the site, but we also have some gene protein analysis, metagenomic, transcriptomic, and utility tools. Those that are in blue are supported for both our bacterial and viral communities. Those that aren't are either bacterial or virus. So like SARS-CoV-2 genome assembly and annotation is a viral tool, or the metagenomic read mapping is bacterial. And these are all screenshots from the site, so you can begin to kind of see what the resource would look like if you're not familiar. Now we're going to hop over to the site and begin to get comfortable with it. So this is our homepage, our landing page. If you type in bvbrc.org, this is where you'll end up. Have a big search bar that you could do a very general search in. So if you were interested in studying influenza, you could just type that in here, and this will show you sort of all of the different data types that we have related to influenza. Now that was pretty vague, but this is a good overview of all of the different data types that you can pull just with a search like that. So taxonomic data or genomes, surveillance data, serology. I'll give you a second to look at that. And feel free to click along with me on your screens or just follow right along. You can see all of those data categories, and so you can click into the category and that'll pull you in there, or you can pull on the specific thing, like if you search influenza with the intention of looking for this hemagglutinin, you could find that structure. So if you want to go back to the home page, you just hit BV-BRC, and here you can see our search tools, our analysis tools, data management, batch access, which is the command line, and API, which I mentioned. If you ever need help, we have documentation for all the tools, and I highly recommend doing our quick start. Got some videos and that'll step you through the resource, and you can search for the help that you're interested in with more detail. And we have our organisms, and these are by bacterial, viral family, and our featured viruses. If you click all viruses, or if you click viruses here, you can get that information about all of the viruses. So since this is more viral specific, that's where we're going to focus today. But I said we had over 11 million viral genomes, and you can see that here. You can see based on the viral families, like I mentioned, and you might not be as familiar with what viral family your virus of interest is in, so I recommend searching for it. But we also have these featured viruses, which are some of the more common ones. We'll click on Ebola virus as an example. Each tab on the taxon view has an overview, the taxonomy. So you'll get the subtaxa of whatever you're clicking on. So these are the subtaxa of Ebola virus. Then you have all of the genomes, and so we have 4,000 Ebola virus genomes. Sometimes it'll take a second to load. Just be patient with that. Any feature data, which going back to the bigger annotations that we have about Ebola virus. So these are features like mature peptides, specific proteins, and you'll see that in the product tag. So like this is a nucleoprotein. And then we have a proteins tab, which has some cool features like switching between the feature tab or getting the FASTA of that feature. And then the structure is where you can start to visualize if you click on the structure. So again, I just clicked here on switch to structure view, and then that'll pull up the PDB structure for that protein. So you can see that here and start to play around with that within the BV-BRC. Then we have domains and motifs and epitopes and experiments. And so these are just all like value adds to the data. And so thinking about it as answering a specific question. So depending on what you're working on and what you're trying to answer, think about how the BV-BRC data can answer those questions for you. And say Ebola virus is too specific, you can always go up the taxonomy by clicking like this, and that will then pull you up the taxonomy and give you more genomes to look at. And again, it might take a second to load. And so we'll go through searches in a bit, but I just wanted to start to get you comfortable with how the data is organized and just little tips like going back up the taxonomy and things like that. All of our tools and services are under this tab. These will allow you to do things like the annotation, assembly, and so on, like I was talking about. Each tool, when you click in on it, has a background and then a quick reference guide and a tutorial, what you start with, and these sort of blocks that change depending on what the service is. And so just fill in all of these, either following the reference guide or the tutorial, and then submit that job. Down here at the bottom, you'll see what jobs you have running. You can also upload data at any point. And so if you had something big that was uploading, that'll have your queue there. The jobs queue is all of the services that you've run, how long they took, when they started, and if they failed. So that's a good place to check in if you're running an experiment. The next thing that I want to talk to you about is workspaces. So we'll go back to the presentation for that. And what is a workspace? And that's your personal dashboard within the BV-BRC. It allows you to add information from your device and internally within the BV-BRC. And there are 3 different options of personal, shared, and public. And so I'm going into an incognito tab because I already have a workspace, but you would register to make a user here. I'm hoping you've all already done this, but if you have not, these are the steps. And make sure you sign up for our mailing list. Get a newsletter coming out soon. So you just fill out this information, set a password, and now you'll have a user ID in the BV-BRC. I mentioned that we have your home directory, public workspaces, and so here you can see all of the workspaces that are shared with me, and this can get a little confusing, so it's important to see what the icon is. So this is a shared workspace, so these are all workspaces. This is the icon for a public workspace. This is the icon for just a workspace that's mine. And you can go into a workspace, and then there are folders. You can copy folders, but you can't share folders. But what you can do is share workspaces. So once you click on a workspace, then you can share, and then that'll add people to be able to edit or view that workspace. So depending on what it is, if you're sharing that information with reviewers, for example, or if you had something that you just wanted to share within your research group, this is a great tool to share that information. And you can upload any sort of data type that you would want. of these options. And so you could share things like papers, CSV files, FASTA data, genomic data, protein data, image data. And if there isn't a file type here, like if you had another file type that you really wanted to be able to share, you can always upload it as unspecified. And so any data can be uploaded. Just select the files on your device and then start upload. Can add folders to your workspace. Just want to tap that quickly, or download the data. We're going to go back to our presentation, and then I'll just touch on searches right after I give you some tips. So you want to make sure to keep your workspace organized. It'll be a gift for your future self. Use folders and name them properly. Principles of that are to be consistent and descriptive, but do whatever will work best for you. And this will just allow you to not have a big mess of information later and allow you to get a good understanding of what the data looks like. The resource starts with 3 folders in your workspace to kind of kick off that organization. So you have genome groups, feature groups, and experiment groups. And so these are just a way to kind of start your organization, but you don't have to use these, and you can add subfolders into these and just move stuff around to keep it organized. We have public workspaces, which we recommend sharing things that are important, limiting the use of this unless it's something that you want to share with the whole BV-BRC community. But that's a good place to find some of our workshops, but the better place to find our workshops is here. So this will give you information about our past workshops, which will allow you to kind of search in there and find anything that might be helpful for you. I'll keep going with some search tips. So searching is the way to really get information out of the BV-BRC. There's 4 main ways of searching, and I'll go through each one. Searching really allows for filtering of the data and refining what we're trying to find out. We have 11 million genomes in the BV-BRC just on the virus side. So filters can help you reduce that data depending on your research question. And it's here where it's important to come into the BV-BRC with a thoughtfully designed question so you can get the most out of the resource. I'm going to go through those 4 options of searches. The first is what I already showed you, which is here where you can be as specific or as non-specific as you want. The second is using the advanced search feature. So this is virus specific, and we'll start with genomes. You can search by keywords here, but I'm gonna use some parameters. So I'm interested in studying influenza A virus, and that'll start filling in. So if I was interested in studying pox viridae pathogens, then that will just start to fill in. And if you hit enter, it'll immediately bring you to that search. So just make sure that you're just clicking on it. You could search by taxon, genome ID, genome name. I'm gonna only search for those with human hosts from Europe from 1960 to 2023. And if you are interested in a specific length, which is important when you're thinking about influenza viruses and the segments, you could do that, but we're going to leave that blank for now. Do segment 4. No subtype or additional criteria, but you could for sure add those in. And then that will just be our sort of starting point. And so if you look at the top, you can see all of the different ways the data has been cut down with those filters. So having the influenza A, human, geographic groups, collection years, and segments, all just ways sort of refining the data. I had touched briefly about filters, but here you can see them. And another thing that you can move around here is the size of this. So depending on your preference, you can make it bigger or smaller. We're actually interested in H3N2 as our subtype. So then you can just click on that. And now we've brought it down to 4,590 genomes. And since we've created this new filter, you can see that those 9,591 genomes that we originally found is actually there. But if you apply a filter, then that will change the group down to this 4,590. And this is good to do if you're interested in filtering the data a bunch of different ways with filters, just to kind of have more control over that. So I'm going to hit apply. And now we only have those 4,590 genomes. You can add additional things to filter on, like the season that they were collected in, or the host name. You can also remove things like and so on. And so this is just a way to give you more control over that data. And similarly, you can change the columns that you see. So we could add a column for segment. Should be 4, so it's not that telling. Or the quality. And so on. Really just thinking about refining that data in relation to a question. If you wanted to put this data in your genome group, if you want all 4,590 of these records, you can click the box here next to genome name, and that'll select everything. And then you can hit group, and this will create a genome group of these influenza viruses. So I'll call this a new group. I'll put it in my default genome groups folder, and then I'll call it. And so this is just how I name it, so I kind of remember those filters that I put in. So then that is that group that I made, and so then I could use that group to run further analysis. So that is one search using the advanced search feature. Another is to start a little bit more broad. So you could start with the Adenoviridae family. Then you can use the taxonomy page to break that data down further. So could do the mastodonvirus genus, and then we get those 22,000 genomes. And now we could use the advanced search to break this down further. So this is another feature that you can use to further filter the data down. Maybe we only want genomes from last year. And so we'll do collection year or the last 2 years, so 2021 to 2022. And that will only pull out of those 22,000 those from the last 2 years, which is 579. And then you can again go into the filters like we've talked about, and you can also reorder your data to be searched by strain or any of these features of the data. And that's kind of just a way. So if we had a bigger data set and you wanted to be able to kind of see how it was structured, it was a good way to look at the size diversity and so on. Another feature that you can use is the search bar up here. This is one that I use often as a starting point. Here you have to spell things correctly to move you in the right direction, but you can get more specific, like searching by the genome strains and so on, or using all term, any all exact term, and any exact term. And so this just means that since I know we have 4 Finchpox viruses, this is searching for all of the terms, meaning fox and poxvirus. But if we did any term, that would be poxvirus or finch, which brings up more genomes and so on, and all exact or any exact. It's just kind of adding more wiggle room, depending on how specific your search is. We'll go through an example for a mysterious outbreak scenario that we did at a workshop. So thinking about a mysterious outbreak in 2015 with an insect host and cases in Africa, and see if there's anything from 1900 to 2015 that can help us find that. So a very broad way we could start is just starting with all of the viral genomes. And so we're not sure what virus it is. So this is a good way to kind of see broadly what the data looks like. And so I said that we're interested from the 1900s to 2015. So we'll do the collection year from 1900 to 2015. And that will break this data down quite aggressively. A smaller number of samples to start, only 1.2 million. I'll just hit apply to see that breakdown. And so another thing that it said was having an insect host. So we'll go to host group of insect. That brings us down to 13,575. Looking only in Africa brings us down to 959. And I think that was all of the features that I wanted to put on this data. So can kind of see what potential viruses this might be. And so this is just like a small example of an outbreak scenario if you're doing very early research to try to understand what there is, but it shows you a good breakdown of the different filters that you can use to search the genomic data. And one thing that's important to notice is that with a certain record, there comes a bunch of data. So we have 959 genomes, but for those 959 genomes, there are over 3 million associated features just with those records. And so proteins, if we apply those filters, that'll be important for those features. Actually, 2,000 proteins. But so you can map between the tabs the genome ID, and this is like the BV-BRC identifier. And so I just clicked on that genome ID, and so this is the genome, this Aedes flavivirus genome. And so we have the genomic data, but then we also have one feature and one protein. And so you can move between tabs for a given genome or a given group of genomes, and that data carries on as you go on. And so that means that when you click on one genome, the view changes from a table to just something like this. So don't be alarmed by that. But so this metadata that's associated with this genome record, like the collection date, the isolation country, the host, is associated within our system with the features and proteins associated with those records, which allows you to have control over that data. And kind of you made a phylogenetic tree later on with the protein data, then you can add the metadata features of country to color it by, for example. We also have genome browsers, but I'll show you that in the SARS-CoV-2 variant tracker, which is another tool that we have in the BV-BRC. The landing page will be updated soon, but the genome browser allows you to see the different features and proteins, class, antibody classes, domains, regions of interest. You can search through this depending if you're studying SARS-CoV-2. Our phylogenetic tree is updated regularly with emerging variants. Sometimes you might just have to load that again. So this tree is all of the variants with their pango lineages, sequences, and then where they're separating, you can scroll in, collapse certain nodes, go to the subtree, so you can see the sequences that in those subtrees, can change the display view. This is using Archaeopteryx. You could download those images for potentially a paper with their use then. You can uncollapse what I just collapsed. can move it around so it gets better for your pictures. Download the data, and I'm going to just reload it to pull everything in. This is a great resource if you're interested in understanding the SARS-CoV-2 variants that are circulating at any point. And see the coloring based on pango lineage. This purple being the EA1, I believe. I can move this bar so that the fills, see what it is. All of these things are changeable, and so you can also change the node fill color. So right now it's by pango, but you could do it by country. And it's pretty messy when it's a big branch, so maybe you could use something like the pango lineage to go to a specific branch, the VOC to a specific subbranch, and then kind of add the colors depending. I will also just quickly show you our YouTube. Here you can see the BV-BRC YouTube page. And we have some instructional videos and tutorials, and webinars and presentations, where this one will end up. I have archived videos from Patrick and IRD and Viper. So that was something that I failed to mention at the beginning. The BV-BRC is a combination of 3 previously existing resources. On the bacterial side, it was Patrick. The viral side, it was IRD and Viper. So these resources merged together to form the BV-BRC. Let me just go to my conclusion. So we went over what is the BV-BRC, created a workspace, searched for data, filtered and curated that data based on a scientific problem, saved our data to a workspace, and set ourselves up for further analysis by putting data into our genome group that allows for further analysis. Here is the whole team that works on the BV-BRC project right now. And we are a NIAID Bioinformatic Resource Center. And just want to open it up for questions, and thank you all for joining me today. As you're maybe forming some questions, just want to point you to where you can always ask for help with the BV-BRC. If you hit contact us and just put in a request here, I or our bacterial outreach coordinator will respond to you quickly or point you towards a developer to help answer any questions. You can also get this contact us form if you have submitted a job that's failed. You can report an issue, and then we can have somebody, one of our people from our software team, see what happened and how we can help you. We definitely want the resource to be as helpful for you all as it can be, so definitely let us know when you have issues. I can't say whether or not we'll have a multiplex primer design service, but what I can say is that we definitely take requests from our user community. So if that's something that you would need for your analysis, I will definitely bring it up that was something that was raised and see if that's something that we can support in the future. I believe that is something that we're open to a discussion about. So I will bring that back to the team and see if I can't get that working in your way. Are you asking about peptides that have been associated with immune responses? Because that's the discussion that we've been having, is kind of labeling influenza peptides that are associated with an immune response. But I'd have to connect you with our team to get a better answer for that. So surface proteins? Do you have a virus of interest? We definitely have people that use the BV-BRC for vaccine design. So we just think about one surface protein that would be common. So we're thinking about a spike protein. That data is housed within our features primarily. So let's see that those spike proteins are there. And we started from source. So if we started from SARS-CoV-2, for example, is a good starting point. I'm just going to filter those from Australia just to break down the data a little bit so we don't have quite as many. But then you can definitely do it with more. It'll just take a little bit longer than me demonstrating it. Going into the features tab. And so here we can filter based on product. And you can search within these. Like surface glycoprotein, but let me just. Yeah, so you can search by anything depending on the virus that you're interested in, like HPV. We do have those. All of these, like surface glycoproteins. Trouble putting down the product here. Sometimes you can do an advanced search by product. Let's call it some minutes here. But so we'll say the surface glycoprotein. Can click on 10 records up to 10,000 and download those genomes and do your analysis on them, or put them in a group and do further analysis using our other tools, align them. All of those could be potentially interesting from a vaccine design perspective. But yeah, we definitely have users using the BV-BRC doing that right now. And you can put together a little bit more direct information if that's something you're interested in. I really appreciate you all coming. Feel free to try to search for whatever data you're interested in. If you have any questions as you're going through all of that, we're here to help and excited to have you using the resource. And yeah, thank you so much for tuning in today. I hope you all have a great rest of your day.